Hello, everyone. My name is Angela Hickey, and I'm a fourth-year PhD student at Stanford University in the lab of Bot. And I'm really excited to have the opportunity to talk to you about my work today, uncovering gut prophage biology using long-read metagenomics. First, I just want to start with an acknowledgement of my co-author on all of this work, Dr. Jakob Borbel, a postdoc in our lab, who's been a fantastic mentor and collaborator on this project, and I wouldn't have been able to do this without him. And I'm really excited to be able to present this work on our behalf. So our lab is interested in studying the human gut microbiome. And when you think of the human gut microbiome, you typically think of the tens of trillions of bacteria that are living in your gut right now. However, this is actually a complex community of different microbes, including the bacteriophages, or the viruses of bacteria, that are actually present in 2 to 1 to their bacterial hosts. So there's twice as many bacteriophages as there are bacteria in your gut right now. However, these have typically been understudied compared to their bacterial counterparts due to technical limitations. However, in recent years, there's been advancements in metagenomic sequencing that has allowed us to start to characterize and catalog the diversity of gut phages that exist in this community. And this has led to relationships between gut phages and human health outcomes, such as IBD, colorectal cancer, diabetes, and Parkinson's disease. However, there's lots of work that needs to be done to understand the relationship between bacteriophages, bacteria, and relating them to the human host. However, in other communities, we know that phages can shape bacterial communities through direct and indirect interactions. So typically when you think of a phage, you likely think of the virulent phages, which inject their genomic DNA into their host. They make many more of these viruses, and then they lyse and burst out of the cell, killing their bacterial host. However, there's also temperate phages, which after injecting their genome into their bacterial host, they use a phage-encoded enzyme to integrate that genome into the host chromosome. And then they can coexist with their host for long periods of time until they're then induced back into this lytic cycle. And we're really interested in these temperate phages because a majority of the phages in the gut are thought to be temperate and have the ability to integrate into their host chromosome. And this presents a lot of interesting indirect interactions onto their bacteria. These integrated prophages can encode genes such as virulence factors and antibiotic resistance that can impact their host bacteria's fitness. And they can also mediate horizontal gene transfer between different bacterial hosts. So we were really interested in understanding how these prophages are interacting with their hosts over time in the human gut microbiome. However, previous studies have faced challenges in being able to look at these kinds of prophage dynamics. And this is because it's really hard to actually assemble viral genomes from metagenomic samples. They typically result in fragmented assembly. From short-read sequencing efforts, we know that it's difficult to assemble repeated regions, and phage genomes have a lot of repeated regions. As well, in mixed samples, there's a lot of phage mosaicism in which regions of different phages look very similar. So this has been difficult in being able to understand not only the host context of these prophages, but also what genes they may be carrying. There's also been few total community studies, which limits our ability to look at bacteria and phages in the same sample. Typically, studies of the virome have used virus-like particle sequencing to look specifically at the viral sequences. However, we want to look at the bacterial host and the viruses in the same sample. And there's also been few longitudinal studies limiting our ability to look at prophage dynamics over time. And lastly, when we want to look at relationships between phages and their hosts, it's been difficult to actually pair these relationships and understand which bacteria or which phages infect which bacteria, which limits our ability to then also do downstream characterization using other wet lab methods. So we were really excited at the potential of using long-read sequencing technology to be able to overcome these challenges. So in order to do this, we generated a long-read longitudinal metagenomic dataset in which we had 6 healthy human donors, which donated stool 2 times across 2 years. And we then took those samples and we did Illumina short-read sequencing, as well as Oxford Nanopore long-read sequencing. We then did de novo assembly of these sequences and then phage annotation using Genomad, Vibrant, and VirSorter, which are standard phage annotation tools. And when we looked to see how many phage regions we found with our different assemblies, here showing just the total number of phage regions for our different assemblies, we found that our short reads actually had many more phage regions compared to our long reads. So here we're looking at our short read assemblies for all of these samples sequenced to a depth of 6 gigabases. However, for our long reads, we had many more gigabases of depth per sample. So we had 30 gigabases-ish per depth for our samples, which is quite deep. So in order to accurately compare these, we subsampled those long reads to 6 gigabases. And as you can see here, there's fewer phage regions that are found. However, when we looked at the total number of integrated phage regions, we found that long reads were better able to capture these prophages compared to short reads. And when we look at the total fraction of phages that are found to be integrated, we see that for both of our long read assemblies, about 60% of all the phages found in these samples were annotated to be integrated, as opposed to the about 5% from our short read assembly. And so we see that our long read assembly really recapitulates the previous findings that a majority of the phages in the gut are temperate. And when we look at a specific example, we can begin to understand why short reads has so many more phage regions annotated. So here we're looking at a long read contig, a section of a long read contig, and we can see that there's phage predictions here from Vibrant, Genomad, and Virusorter, in which we see that there's an integrated prophage on this contig. When we look then at the short read contigs mapped against this long read contig, we can see that each of these gray boxes represents a different short read contig, and that it's a very fragmented assembly in the short reads. And then when we look at our short read phage prediction down here, we see that there's many phage regions that are annotated, or phage contigs that are annotated here, that actually represent a single integrated phage. And from our long reads, we're able to actually know that all of those phage regions are connected together. And when we look at the gene content of this region, we see that we find things such as phage tail proteins, other phage proteins, as well as an encoded integrase, which we would expect since we find this integrated into a host chromosome. And more importantly, because this is now assembled in its host context, we can look at the surrounding region to identify which host this phage is infecting. And we find that this is an Ruminococcus lactis host, something that was very difficult to do before. And so now with this, we can use long-read sequencing technology to begin to understand prophages and their hosts and that interaction over time. Specifically, we were interested in understanding how stable prophage integration is in the gut over time, as well as what are the integration strategies of gut prophages. So first to start, we wanted to look at the stability of this integration over our 2-year time course. So again, using this Ruminococcus lactis host example, we can look at our 2 time points at the read coverage across this region to look at the dynamics. Here, we're looking at read coverage from our first time point here in light blue at the top, and then our second time point reads here in the dark blue. And what you can see is that there's pretty even coverage across the host chromosome, but when we look specifically at the phage region, we find that at the second time point, there are very few, basically no reads that map to this phage region. And so we can see that this phage has been lost from this host over the course of 2 years, while its host remains. And we were interested in looking at this for all of our samples. So here we are looking at the percentage of phages per our different participants and looking at the stability of this integration. So here we found that there are phages that are stably integrated into hosts that are either gained or lost over this 2 years, which represents just normal bacterial turnover. While we also found phages that are stably integrated into persistent hosts. But what's interesting is that we find about 5% of these phages in each participant that are dynamically gained or lost. So similar to this Ruminococcus lactis example, in which the host is still present after 2 years, but the phage region has either been gained or lost. And we also find that there are phages that are found in multiple host contexts. So using this, we can now look at how stable prophage integration is over time, finding that a majority of phages are stably integrated across 2 years, but that about 5% are actively gained or lost from bacterial hosts that are persistent over that time. And what's really great too is when we look at the structural variants that we find where phages are then lost or gained from a host, we can use that to determine the exact phage boundaries, which allows us to look at all of the genes that are actually a part of that phage. Next, we were interested in looking at what the integration strategies are of gut prophages. So this was work that was done with a fantastic rotation student in our lab, Daniel Chang, in which he looked at our phages that had this exact boundary evidence to look for which phage integration enzymes they encoded. So he looked for canonical phage integrases such as tyrosine integrases, serine integrases, and DDE integrases, and interestingly found a handful of phages that are found integrated but don't seem to encode one of these canonical integrases. And when he looked at these even closer, he found that there were 22 phages that had a really similar shared genome organization, in which the phage genome was bound by two IS30 elements. And IS30 elements are bacterially derived insertion sequences, which are selfish genetic elements. So they typically encode these inverted repeat regions, which surround an encoded transposase, which can then move this IS sequence around the genome. However, there are also IS elements that can flank a piece of DNA, creating what's called a composite transposon, in which this IS encoded transposase can actually mobilize this entire region the same way it would mobilize itself. And so we were really excited at the potential for these phages to use these IS30s as their integration and excision machinery. And these we have affectionately named as IScream phages as an homage to the Cree recombinase, which has been very useful in molecular biology and was also derived from a phage genome. And when we looked at these IScream phages in our samples, we wanted to look at if they had any shared genome organization. And here we're looking at a synteny plot of the IScream phages from our sample. And we see that on the left, we have the IS30 element found in all of these phages, as well as phage genes for packaging, as well as lysis from their host. And interestingly, there are some very tight clusters of these IScream phages where we see that they're shared between the same individual across the two time points, as we would maybe expect, but also that there are really similar clusters here with an ANI greater than 95% that are found in different individuals. And what's interesting is if we look at the bacterial host of these IScream phages, which hosts that they're integrated into, we find that they're found in really distantly related hosts. So here they're part of the same taxonomic order, but different taxonomic families. And we can also find another example of these kind of clusters in which we have, again, different bacterial hosts for very similar IScream phages, in which they're found in the same taxonomic family, but different taxonomic genus. And we were really interested in this since a lot of phages are thought to have a very narrow host range, either infecting the same species or even sometimes a specific strain of a species. And so we wanted to look at this across our other phages. So we looked at phage clusters of all the phages found from our different samples. And we wanted to know what was the broadest host taxonomic level that we saw these phages integrated into. And what we find is that a majority of phages are found restricted to a single species. We only see them integrated into one species. However, there are others that have a much broader host range, like the ones we see with IScream phages, in which they're members of the same taxonomic order, but found in different taxonomic families. And here's another example of one of those in which they're found in the same order, here Bacteroidalis, but found in different taxonomic families. And here we can see the phage region annotated in teal, as well as the synteny between these phage regions. And we see that there's very even coverage between the phage and the host, indicating that this is evidence of this very broad host range. And so while we're looking at the integration strategies of gut prophages, we're able to annotate a new class of IS-bound prophages, here called IScream phages. And while a majority of the phages in our sample are found restricted to a single species, we do provide evidence that there are broad host range phages that are restricted to the order level existing in the gut. And this really represents a lower bound of these types of broad host range phages, which may be present in the gut. So just to summarize what I've showed you today, we see that long reads can unlock novel phage biology and enable us to actually capture the prophages that are present in the gut. We also find that by looking at stability over time, that about 5% of phages are dynamically gained or lost across a time period of about 2 years. We also describe a novel class of IS-bound prophages, IScream phages, that may have co-opted bacterial IS elements for integration and excision machinery, as well as evaluating host range in the gut environment. And we provide evidence for a broader host range than has been previously reported, in which phages are restricted to the order taxonomic level, but found in different families. And if you're interested in any more of this work, please check out our preprint now on bioRxiv. And lastly, now we can have a better understanding of how prophages interact with their hosts over time in the human gut, really enabled by this technology. And with that, I'd love to thank my lab and everybody who contributed to this work, especially my co-author, Dr. Jakob Warbel, as well as our collaborators, my thesis committee, and our funding sources. Thank you.